Good evening and welcome to the inaugural lecture of Professor Jim Bown. Jim began his relationship with Abertay in 1992 as an undergraduate student in computing and went on to doctoral study with the Scottish Crop Research Institute, now the James Hutton Institute. Following this, he became a founding member of Abertay's Symbios Research Centre and began his lectureship at Abertay in 1999. He's published his work on plant and fungal community dynamics in Nature, the Proceedings of the Royal Society and Royal Society Interface. To support this research and his more recent research activities, he secured over 1.5 million in research funding. Jim then led the cross-campus research agenda, white space research, as we call it, that combines computer games, art, psychology, sociology, computing, and environmental sciences to provide solutions to the problems facing industry. This research activity helped secure funding for Abertay to establish the National Centre for Excellence in Computer Games Education and the Prototype Fund to support small digital business. At the same time, Jim was a partner on a project called Cosmos, a large EPSRC-funded interdisciplinary project that provides both a model development process and a massively parallel distributed simulation environment to support complex systems research. Jim has been appointed to the chair in systems biology and since 2009 has moved into the field of cancer systems biology. His work exploits techniques from ecological modelling, interactive visualization, through computer games technologies and massively parallel computing to understand cancer cell signalling and tumour growth processes. Jim will present his inaugural lecture on his latest research here this evening. Thank you.
Good evening. Thank you, Nigel. Thank you for coming. Serious games for serious science, so this is going to be quite a serious lecture. The topic I'm going to talk about is one of the most serious diseases on the planet. 1 in 3 people will develop cancer in their lifetime. That's you or the person sat to your left or the right. There's a diagnosis made of a new patient with cancer every 2 minutes. 30 in the duration of this lecture, assuming I don't overrun. According to Cancer Research UK, it's the UK's biggest fear. It's not job loss, it's not debt. And thank you, Robin, for the image of fear there. Over 50% of cancer in the UK is localised around 4 particular conditions: breast cancer, lung cancer, prostate and colorectal. But there's a very long tail of other cancers, smaller but equally difficult to treat. 1 in 4 people will die from cancer, and 3/4 of those are aged over 65. But only 3/4. The 5-year survival rate is currently 50%. It's increasing slowly, so things are getting better, but there's a very long way to go. A study by the World Health Organization and the International Agency for Research on Cancer surveyed 2008. There were 12.7 million new cases per annum and 7.6 million deaths. And this is a growing problem. By 2030, that number is going to be 22 million or more new cases. This is a very, very pervasive issue. In terms of the financial burden, if you like, the pressure that cancer generates on the healthcare provision, worldwide in 2008, $895 billion US. And that's increasing year on year. Now, partly that's down to aging, partly that's down to population size, partly that's down to new technology simply being more expensive because we're trying to do more, and there's an overall expectation of an increase in healthcare spend. That's what people simply expect as things develop. In terms of the total spend, what we've got is about 5% of the total healthcare budget. That's quite a lot. And one of the difficulties is, how do you know in a very financially constrained environment that you're spending that money well? Cancer drugs are very expensive to develop, they're very expensive to treat patients with. NHS, other health organisations have only got a limited amount of money. How do they best allocate? And one of the ideas that's been developing over the last probably couple of decades really is the notion of a biomarker, a biological indicator that would suggest whether you as an individual would be likely to benefit from a particular course of treatment or not. And that can help stratify that investment. If your cancer has particular biomarkers that would suggest particular drug interventions are more likely to work. That's at least the theory. To explain a little bit about biomarkers, I need to do some cell biology, not my field strictly, in about a minute as well. So what we have here is a picture of a cell. This is the cell wall, the cell membrane. The big purple thing at the bottom there, that's the nucleus, and the signal receptors up top. These signal receptors listen to outside stimuli, what's going on in the wider body, and respond. And they respond through these particular, they're called signalling pathways. So signals are received by this cell from the environment that might encourage cell growth, it might encourage cell division, it might encourage cell death. And they propagate down through this network, a little bit like a telecommunications network, down into the nucleus, passing instructions down, but also feeding instructions back up as well. So this particular pathway here is one example of the survival-death pathway. On the other side, we've got cell division, where cells divide from one into two new cells. Now, of course, this is a very simplified diagram of this signalling pathway, and so is that. These are enormously complex systems, and that complexity attracts a particular challenge. Cancer dysregulates this kind of pathway, and it dysregulates that pathway to promote particular behaviours in cells. So cells don't die when they should. They divide and so multiply far more frequently than they should. They ignore the external signals to stop growing. For example, across the top, I don't have time to really go through in any detail here, but cancer itself finds multiple ways of disrupting this complex signalling system to affect cells that either live for a very long time, if not forever, divide far more frequently, and don't stop growing when they should. But that's okay because there's drugs. Chemotherapy works by targeting cells when they're in key states. So these cells have a particular life cycle. They go through a particular series of processes. And when they're dividing, for example, they're reasonably vulnerable to shocks in the system. And this is what the drugs try and do. They try and perturb what the cell trajectory is going to do, where that cell is trying to get to. One of the papers that I read recently from one of my collaborators, one clinical trial drug used to treat ovarian cancer, it's effective in only 7.5% of patients. And that's pre-selected patients, so after you've done your biomarker. to say this drug might work. It only works on 7.5%. And then you get a problem. You get an initial response from a lot of patients, but then for some patients, the drug stops working. And it's very, very difficult to work out quite what that is, but it's called drug resistance. It's resistant cells. They're not reacting to the drug. Now, you can drill down into data and try and find biomarkers that would indicate whether a patient would be resistant or not to a particular drug intervention. That's only half the story. That's a very phenomenological understanding. You're only understanding that as a picture. What about the underlying mechanism, the gearing that happens behind the statistics that say this is a patient likely to be resistant to this particular drug or likely to be susceptible? And it's this interplay between this kind of data that tells you statistically whether a patient's likely to react and these underlying mechanisms that are going on inside that cell signalling. And so some of the work we've been doing is trying to understand not just biomarkers for resistance, but mechanisms for resistance. In order to have a, without getting too bogged down into cancer, I'm sure you recognise what that is. This is the London Underground. What the drugs do, if you think about these train lines as the signalling pathways, right? The trains go round and round the network. What the drugs try and do is target particular areas in that network to disrupt the cell from its particular behaviour. And it's a little bit like having a fault on the line, right? Could be targeted or not. But of course, if you've been to London, it's not difficult if one line is down to get from A to B. It might take you a little bit longer, but it's not difficult to make that journey. You just have to reroute a little bit. And actually the cell's very good at that. If you knock out one way of doing something, it will find another route, another mechanism to overcome the block that you've put in to achieve what it's trying to do anyway. And so we come to combination therapies. Therapies not based on a single drug, but on a series of drugs given at different dosages at different times. So you might give a dose of a drug for a short period of time for a patient and then move them on to another treatment. So you're perturbing this network into one state and then treating it with the drug. And that's a little bit like trying to get around the network when there's more gone wrong. And so this is the kind of this is the sort of metaphor for understanding combination therapy. Of course, I said these networks were complicated. In fact, they've got complex behaviours. And I just want to show this is some data from the lab. Simon, you'll recognise these graphs, I'm sure. I've simplified it out. These are two particular measurables inside this signalling network. We've got, on the left-hand side, one measurable, and we've got two different conditions, either with a drug or without a drug. And you can see the effect of the drug is to increase this particular signalling. On the right-hand side, we've got another output from the same drug. So here we've got output from the signalling network with a drug, and here's a different output and a different response. The drug doesn't do that much. It brings the level of activity down. We use a different drug on the same output stream, and you get another increase in activity. We use a different drug on the same output stream, and we get a decrease. Now, one of my colleagues used to say that these are graphs without a y-axis. And what that means is that the values here are kind of relative. It's more about the direction. It's not necessarily the size of the shift. It's the way that it's going. So here, drug A and drug B both push this signal up. And here, one pushes it down and the other one up. So if we add both together, what we get on this side is not up at all. If we add the combination of A and B, we get almost no response. There's no difference. If we add the combination here, we get a very dramatic response, but completely unexpected, I would suggest. And that's an indicator of the complexities that are going on inside these cells. In order to try and understand those complexities, we have to turn to a mix of systematic experiment and computation. And this is the field of systems biology, specifically cancer systems biology. And there you're trying to use models to understand the functioning of these cell signal networks. So what we've got here is exactly that same signaling network that I showed you previously. We've got the survival pathway, we've got the proliferation pathway, these cascades of interactions structured around these signal receptors down into the nucleus. We can model that. And so we effectively copy that topology, that network shape in a computer, in a mathematical computational model. And we can use this model to perform experiments that would be either too expensive or too difficult to do in the real world. We can help cut costs down or do things that are not strictly possible. So I spoke earlier about biomarkers. One observed biomarker, 2004, is an element called P10. It doesn't matter what it is, but the fact is that if you're a low P10 expression patient, your survival chances, this is a survival curve, likelihood of survival over months, you're far less likely to survive this particular cancer than if you're high P10. So phenomenologically, P10 might be a good biomarker. But in terms of the signaling network, what's going on? Why should that be a good biomarker? What about the mechanism? And so we extract the region of our modelling framework that is associated with P10 and what it's talking to in its nearest neighbourhood. Through a very extensive modelling approach, there was a sophisticated analysis done that determined effectively a volume control for sensitivity to a drug or resistance. And it was all about a particular ratio of 3 things. So the argument is that if this is the control parameter, this particular ratio, and if you're working with P10 that's in low or high, what you need to do is to manipulate the other part of the term to switch between sensitivity and resistance. So if you can't shift P10 around, if that's what the patient is, then you go for example, PI3K. You can make that into a sensitive function rather than resistant because of the relative balance of those 2 particular activities. And so you essentially have this spanner, which is our drug, the thing that we're trying to manipulate into the system to get a sensitive response to, and 2 other spanners. One is a drug around P10, which puts it in a high or low state to simulate cancer mutation, that graph observed in the paper. And the other spanner is our other side of the equation, this PI3K term. We could have equally done it with AKT, but that's just the particular diagram. And so by manipulating those 2 spanners, the relative balance, you can shift the patient from resistant to a drug to sensitive to a drug, is at least what's coming out of the model. Well, does it work? Well, the dark blue here is the model predictions, and the light blue is the experiment. And what we've got is a particular system that you stimulate with this growth factor, and so we've normalized, we've set the maximum to the maximum that we observed. And so you get a response from the growth factor. We add in the growth inhibitor, the thing that's meant to stop growth, and you get a decrease Well, that's good because that means the inhibitor works. We then introduce, with a different drug, this P10 mutation to make that low. We get a resistant model again, both in the model and in the experimental system, and we can restore sensitivity to the drug by adjusting the bottom half of that equation, the bottom half of that ratio. So you can put it back into sensitivity. So it responds then to the growth inhibition. So everything's great. It all works fine. So you can control growth inhibitor effect by different perturbations to that particular ratio. Well, that's all very well, but that's in a Petri dish, not in a patient. And that actually took an enormous amount of mathematical analysis by one of the modelling guys. What I want to talk on, with that cancer biology, as I've tried to cover it in 20 minutes in the sort of world of cancer, I want to talk a little bit about 4 things that we've been doing to try and move this field forward on top of that foundation. And one of them, the first one I want to show, is about putting the model in the right hands. As I said previously, it was the modeler that did that analysis and determined that relationship. Now, this particular individual knows an awful lot about cancer, but a lot of modelers don't. Should it be the modeler? Now, what happens here is that we as modelers listen to our biological collaborators. They draw the network on the board, they tell us what it is, and we copy it down, and we make a network out of it. But we do something else. Build a load of equations that are effectively the timetable for this train network. So we build a load of equations that describe how those signals cascade at different times. We then do the easy bit, which is to code it up. And then we run simulations and we show them results and everything's brilliant. And what we show our biology collaborators is the very network that they gave us in the first place. So if we've copied it down right, then everything's fine. And when they ask about the results and how did you get them, oh, well, that's all down to the differential equations, and there's 60 of them. And I remember Nicholas being particularly disappointed when I pointed out that I wasn't very good at reading 60 differential equations. It's quite hard. There's a lot of terms going on here. I'm not convinced anybody actually follows what's really going on over the kind of timescales that we're talking about. So you've got, on the one hand, this really simple relationship with what we were given. And on the other hand, this impenetrable mass of symbols. How do you know what this model's doing? And this is where the serious games bit comes in. Now, serious games, it's a sort of catchphrase. It's just a way of exploiting game technology in non-entertainment, i.e. serious environments. Now, we've done a little bit of this before as a university. One example is the work of John Isaacs in the SAVE group led by David Blackwood. This is, sorry, I'll jump back a bit. This is the architect's map of the redevelopment work down at the waterfront. Architects are really good at looking at these pictures and seeing in their head what it's going to look like. I hope. I suspect members of the public are not so good at looking at a map like that and saying, right, I see what that building's going to look like because I know what it's made of, I know how long it's going to take to build, I know what the energy requirements are. And so what we've done, what John and David and colleagues have done, is built effectively a game engine that allows you to import these buildings in, the architect's models, and bring it to life. So you can see, as non-architect experts, what this thing's going to look like. You can zoom in, you can have traffic models, you can do all sorts. You can even change the weather like it is today and have it rain. My brother works in flood defence. We may be needing you. That's just coincidence. The flooding was already in the movie. What we've got here is, if you like, our way of putting the model, that complicated set of differential equations, in the hands of the biologists, the people that actually know the underlying biology. So what we're going to show you in this first movie is essentially the model running. Now that's okay, that's an animation. That shows the sequence of events, that the growth factors come in, the signals propagate down. Now the nodes, the clumps in this network are related to the nodes in the previous signaling network, and the size depends on the level of activity. The thickness of the lines is about the extent of interchange, so you see the biology that's going on in a picture from the differential equations. Now this is good, because if we got this wrong, they can say, but that's not what we meant at all. That reaction goes the other way. You can't see that in the differential equations, at least easily. This brings that to life. But that's not the real strength of this. What we can do through this same interface is add drugs in. So through this little pop-up box, you can choose from a drop-down list, and you can add drugs of a particular concentration at a particular time, actually as many as you like. You can build a cocktail of drugs, this combination therapy. Run the model, and what you see is red representing increases in activity and blue representing decreases in activity. Now this red and blue, this particular drug, this was the growth inhibitor. So this is going to reduce a lot of the activity in the signaling network. And you can see where, not everywhere, and not everywhere differentially. In fact, some areas are upregulated by that particular treatment. At least we can get confidence, because that might make some sense that this is high and this is low. What you can look at on a node-by-node basis is comparison with one and another. You can drill into exactly what's going on. You can wind time backwards and forwards. You can add another drug, and everything we add in, it recalculates and shows you the diagram again, as a movie. Not only that, but you can add in stuff you don't know about. So if you don't have a drug, but you think, what would happen if I changed that value and brought it down by a half? Big list of everything in the model. You can just change it and see. So it then becomes not just a diagnostic, but a potential drug discovery tool. And of course, as Margaret was quick to point out, this isn't just for drugs. This is any network. It could be a food supply chain. It could be a transport network. Anything where you've got interplay between lots and lots of different parts. So we think this is going to be very useful. It's not finished. I always say it's not finished to David. All right, what do we do next? Well, firstly, we've got a quick patent file just to protect us, get a marker in the sand. How do we get the interaction right? And you probably can't see this unless you're one of the lucky ones. I say lucky. Sat at the very front, that's somebody getting shot. This was from Paul Robertson and the work he did on developing a serious game for firearms training. But one of the things Paul did was evaluate very rigorously whether that interface was useful to the experts. Well, we're going to draw on that. I say we, Paul. We're going to have a look at task performance with experts and see if this is the right interface. Is it blue and red? Is it drop-down boxes? We've got to get the interface on this right. There's a bit of a technical problem. There's an issue about portability because it's quite tightly locked into our current computation. But I'm not too worried about that. And there's some other stuff that doesn't quite make sense in my head. Another challenge from the successful modelling I showed you, this signal network modelling, is the time it takes to make one of these models. It's not weeks. It's kind of months. There's a lot goes in there. And if you add in something new, you've got to play around with a lot of it and recalibrate, and it's quite painful. So, Michael Idowu, who's sat in the audience, hello Michael, has developed a technique that takes time series data like we get from these signaling networks, and does some complicated analysis on it, and actually makes a network reconstruction. Now, the important point here is that we don't show this model that network. We only show the model the data. We do the analysis, reconstruct the network, and compare with the known but unseen network. So it's a kind of guessing game. How do you work out what's there? And the approach that he's developed, filed a patent on, driving this forward as inventor, can recover about 85% of the links. Picks up a few extra ones, but that's development. So this can be a very fast first guide. Michael's already applied this to time series data from that signaling model I showed you, and his networks show that you can spot the odd one out. You can see the sensitive, you can see the resistant networks, which is kind of useful. And more recently, he's been working with Hillel up in the audience there on real wet data and all of the noise that comes with that, and again has prompted a new set of experiments that borne out to be true, and that's currently being written up. We're moving away from cells towards tissue, and I'll come back to why we want to move towards tissue shortly. But in terms of tissue itself, we've been looking at how to quantify tissue structure. A lot of diagnosis is done by holding up an image against the window and saying, 6 months, or we can operate on that. You know, and that's actually as good as anything we've got at the moment. What we've tried to do here, with the advent of MRI technology, other 3D scanning type techniques, is develop a way of quantifying three-dimensional tissue. And actually, if you were at Wilfred's inaugural lecture, you'll recognise this movie, because this is soil. This is a cube of soil, and in a moment the video is going to release the internal pore structure. And what Wilfred's really interested in is quantifying that pore structure for reasons to do with soil function. But that's the holes. What we're interested in is how tumours fill the available space. So it's exactly the same issue, just the other way round. We're not interested in the hole, we're interested in the solid. So all we need to do is use the algorithms that symbiotes have already developed, Minkowski functionals, which are well known to characterize 3D structures, and change a 0 to a 1. That's it. And run it over the data. We're not looking for the gap, we're looking for the fill. It's just 0 or 1, simple enough. What we found when we plotted one of these quantifiers against another is for untreated tumours, we can't separate them out. But for treated tumours, we can see a difference. We can stratify treated and untreated in terms of these values. Tumours are graded 1, 2, and 3. 1, not so bad. 3, very bad. We looked at could we predict the grade from these measurables? Sorry, I've gone too far. These particular things. I should have said, one of them is tumour volume, one of them is the surface area of a thing. One is how sort of knobbly the tumour is, and another one is how many spikes are coming off. And just from those measurables, could we determine what the grade was that the pathologist gave it? And it turned out, for grade 1, so for not serious tumours, we could do it. I know the numbers are small, but that's the data we had. We could do it all the time. For grade 2, we could do it all the time and get it right. For grade 3, for the untreated tumours, we had no problem. We got almost all of them right. That's a pretty good result. For the treated tumours, to our dismay, we got them all wrong, or very nearly. But there's a reason for that. It's because in their morphology, their shape, they look like grade 1 tumours because they've been treated with a drug. It's the drug effect. And we're picking that up. And we've published this quite recently. I want to finish off with something that might be a bit more ambitious. There is a big problem in this field, and it's about linking scales. It's about linking cell signalling, the stuff I showed you about this signalling network, up to tissue. The tissue issue. One way of looking at tissue is that it's completely homogeneous. That just means it's all the same. Of course it's not all the same, right? Because there's stuff in it. It's not just cells, be they cancerous or otherwise. There's blood and there's other bits and pieces. There's the immune system acting in there as well. It's actually quite a murky environment. So it's not all the same. But not only that, the cells are different. When you chop through some tumour, if you chop through a tumour and you look at, for example, one of these signalling values, in this case it's a drug target, you get an incredible amount of variation. If your drug target, which is going to damage these cells and perhaps kill them off, is focusing on this particular signal, you'll probably get all the red ones, but what about the blue ones? All you've done is just given them some space to grow. So you've applied this really toxic drug, upset the patient, these things are not nice, and you've killed off some cells, but it only takes one cell and this stuff comes back. That's the problem. It's this heterogeneity that's the big worry. Now, Nia and Bill Sampson down the front will recognise that particular image. During my PhD and previously I spent a long time working on fungi, looking at the importance of how things arrange in space, looking at plants and how things arrange in space and time, so the time dynamics of these things, how things change over time in space. We've done some other work on fungi looking at really the big issue in ecology. How can you relate the stuff you can measure to the stuff you can see? Processes that you can measure what's going on to the bigger structures, the patterns that come from those processes. And then the difference between looking at interspecies, so between species comparisons, and intraspecies, within species. And it's this intraspecies is as important as between species. So we've got quite a framework there for looking at the ecology of a tumour. We've got 10 years of track record in this. The essential problem is to get from a cell with its 60 differential equations or all those pathways up to the tissue scale. And what you mustn't do is make cell soup. What you can't have is just one big cell or a big lump of tissue where all the cells are the same, because that will tell you nothing useful, because you're losing that variation. So, my Man on the Moon project, I think everyone should have one. 1 million individual cells, representing 6 months of biology time. All to be done in 1 week of simulation. That's pretty ambitious, but we think we know how to do it. Well, I'm going to say that now, aren't I? Firstly, we don't do cell soup. We build our simulations out of loads and loads of individual cells that are potentially all different. We've got a problem. We can't run that on a single machine because we'll kill it. There's only so much simulation you can do within one machine. It's only got so much memory. And these are problems that will go beyond machines. But that's okay, because Adam Sampson and myself have been involved in a thing called Cosmos. The link between Cosmos and Man on the Moon, there is none, they're completely separate entities. But it's a project, EPSRC, with the universities of York and Kent. And what Cosmos has been doing, among other things, is harnessing the power of parallel programming on multi-core devices. This is a contemporary graphics card. Games have driven up graphics cards such that they're now large multi-core computational resources. Well, let's use them, because actually they're quite cheap. This is a PS3 processor. Bit of a pain to program, but again, quite cheap. So can we use this stuff? Well, yeah, we can. Cosmos has developed ways of distributing code across machines, which can be run on the cluster or indeed the cloud. How does it work? Well, for example, this isn't cancer, this is blood clotting. This was a project called Tuna, Theory Underpinning Nanotech Assemblers. What we have here is a blood vessel and platelets, individual plates that join together when you make a wound. So if you wound the blood vessel itself sends out a bit of a signal saying, I've got a bit of a hole here. Can you join together and help me fix it? So this is the cutting tool. It's all done in simulation. You cut a hole in the side of the thing, in the side of the blood vessel. The healing factor leaks out. And I don't know whether you can see it up the back or not, but certainly down the front, you end up with slightly larger clumps. The clumps start sticking together, and that's the only behaviour. All the individual platelets are doing is, if you come into contact with the green stuff, get a bit more sticky. And over time, that will actually close the hole over. But it's a big problem here, and it's one of scale. This is as much as you could run in a reasonable time frame on a single machine. But the relative size of the blood vessel compared to the platelets is way out. You see it starting to close the hole over. It should look more like that. That's the relative size. And the size matters because you get different behaviours at different scales. But you can't run that on one machine. But through Cosmos, that's okay, because we can run it on 18. We just line them up. So we can run very, very, in fact, arguably, I'll use the word loosely, infinitely large, depending on how much money we've got, resources. We can run infinitely sized simulations. So this next slide here is the same algorithm, but tweaked. It's a different visualization, and actually this is running in real time. The other one took a little while to compute, about a morning. This one's running in real time, but it's the same thing. You can see the clots. The important thing here is inside every single one of these platelets is effectively a signaling network, and they could all be different. We can put different networks inside each one. Now, when we start distributing across multiple computers and from one machine to another over a network, we've got a problem. We've got 3 computers in this room. 1, 2, 3. You guys can talk very quickly to each other. You guys can talk very quickly to each other. But this edge is really slow, relatively, because you're going through a network cable. This edge is really slow. Well, it turns out that games have already solved this problem. You recognise that? Right? Call of Duty. Have I got that right? Call of Duty, good. This is an online multiplayer game. And it really, really matters in this game if this guy shoots at that player there. But because of network latency, this player is able to jump just a couple of feet to the side, and so the shot misses, even though it was a perfectly aimed shot from this perspective. That leads to a really frustrating gameplay experience because you have no control over what's going on. It's an issue called dead reckoning. And there are algorithms from games that can help solve the dead reckoning problems that we face on the interface when things bang into each other. So not on one machine, but over a network. And how do you deal with that latency? They've worked it out. So, we do have a shortcoming in what I've said. I think we've got good biology in our model. I think scale isn't a problem either. What we don't have is a very good model of physics. But that's okay. A new collaboration with Tim Newman from Dundee Life Sciences. He's a physicist who's moved from ecology into cancer because it's interesting, has a really nice model of cells and how they grow physically, not biologically, physically, in physical space. And we've got that simulation, and Adam has recoded it using that same kind of technology. And you can see this is one cell becomes 2 cells, it'll become 4 cells in a moment, it'll become, because these cells are dividing when they reach a critical stage. So what we've got is the real cell, our biological model of the cell, in which we can put our signaling network. So we put the signaling network in the cell. We put the physics in the cell. We've got the biology already. We put the physics in the cell. Cell-cell interactions and cell growth. We do the smart multi-core computing to get very large scales. And we can look at heterogeneous tissue. Tissue made up of different cells. We then turn to colleagues who know about spatial statistics to see how you compare our modelled tissues with real tissues. And because there's mechanisms inside our model, we can begin to understand the processes that generate structures that look like this, rather than nice round tumours. How does that happen? It only happens from heterogeneous tissue. Now I've got one last movie to show, hopefully. And this is it's only because it's quite big and my machine pauses slightly. I did this earlier, it was fine. Press the button again, still nothing. There we go. This is the kind of scale that I'm talking about. We've got about 250,000 cells. Now, there's no model under this. This is just an artist's impression. And I thank Ryan Locke in the audience there and Chris McCready that I can't see for doing the artwork and the games technology respectively. But this is a games engine running a very large simulation. In the middle of this bundle of cells, this 250,000 cells, is a tumour. It's a tumour that's got some yellow cells and some red cells. We're going to get quite close to that tumour and apply a drug. But only one of the cell types is sensitive to the drug. The other one's resistant. So we apply the drug, the orangey ones get knocked out, but the red ones begin to multiply because there's more space. This is what we're able to build. Now, I have presented this to various audiences, and I've had different responses. The biologists normally go, fantastic. The modelers normally go, you must be mad. Because you're never going to get the data to profile all those cells, to characterize that variability. Well, here's the thing. Went to a talk in May. Gary Nolan from Stanford. He's able to characterize individual cells in 45 dimensions. So inside that signaling network of all those blobs, each one of those at 1,000 cells a second. I don't think anyone else is going to be in a good position to deal with that data in this particular way. I think if we can do this, we're ahead of the curve. And that's a good place to be in this field. By combining this games technology that lets you see what on earth is going on, because this is too complicated, state-of-the-art experimental systems, really solid cell physics, good biological modelling, and a little bit of imagination. My next slide says summary, because in my dry run rehearsal I forgot to do the summary, but I've just done the summary. That was it. So it remains for me to acknowledge, firstly, our primary funders, at least in Abertay, the Northwood Trust, and also SICSA, the Scottish Informatics and Computer Science Alliance. Acknowledgements to our collaborators outside of Abertay, St Andrews, Edinburgh, Dundee, York and Kent. These are people we're bringing to this consortium, and you can only do this kind of work through consortia-based approaches. And as for Abertay, I struggled with how to do this slide because there's so many people that I worked with over all this time. If I've missed you off, that was an oversight. I did try and get everybody. So thank you very much.
Thank you, Jim. I'm still reading, right?
You're not there.
Ran out of credit. It's my great pleasure to thank Jim on behalf of the Vice-Chancellor, university staff, honoured guests and members of the public who have joined us here this evening. I'm also very pleased that Jim's family and friends are able to join him in tonight's celebration. Jim's talk fascinated me at many different levels, and I've heard bits of this story over the years, okay, and it was really nice to see it all kind of joined together. I think those of us who have worked alongside Jim, and there's many of them in the audience now, know that what we've heard this evening just gives us just a hint, the tip of the iceberg of what Jim contributes to the intellectual life of Abertay and actually beyond. Okay? As well as the actual results of his research work, what Jim has done is developed a particular way to pursue and advance scientific curiosity. And I think that's the contribution to our, well, certainly to my intellectual life that I've gained from him. That approach has substantially affected a large part of the research culture here at Abertay and has opened doors for external research collaborations of significance. Jim's generosity showed in his acknowledgements, oh, they've stopped now, okay, and his expressions of gratitude to individuals as he proceeded through his talk. But Jim, I believe that many in your audience tonight feel that they should be thanking you for the positive impact you've had on their individual development and their work, whether it's through your insights and research, how to conduct research, through your teaching, through your wise counsel, and actually through your humanity. Tonight I've enjoyed learning and gaining a deeper understanding of the working of science, but the evening is also a celebration of Jim's achievement. Jim, I'll end by thanking you again and joining with the audience in congratulating you on that achievement. So thank you, Jim.